Hello guys, welcome to BIOGURU. Today we will discuss some questions of molecular basis of inheritance. So objective type questions level 1. So the number of nucleotides in 5174 bacteriophage is 1386, 3586, 2586 or 5286. Yes, it is 1386. The number of nucleotides base pairs in lambda phage is 50286, 48502, 20585 or 30585. Yes, it is 48502. That's good. Base pairs in haploid content of human DNA is 2.3 into 10 power 6, 1.3 into 10 power 6, 3.3 into 10 power 9 and 4.3 into 10 power 9. Yes, it is 3.3 into 10 power 9. That's good. Common pyrimidine present in both DNA and RNA is cytosine, guanine, adenine or thymine. Yes, it is cytosine. Nuclein term was given by Mischer, Watson, Crick or Seck. Yeah, it is Mischer. Chemical nature of DNA was studied by Chargaff, Watson, Crick, Seck, yeah, Chargaff. Adenine pairs with thymine by 2 hydrogen bonds, 3 hydrogen bonds, 4 hydrogen bonds or 5 hydrogen bonds. Yes, it is 2 hydrogen bonds. Uniform distance between two strands of DNA 20 nanometer, 80 nanometer, 22 nanometer or 20 angstrom. Yes, it is 20 angstrom. The pitch coiler helix of the DNA helix is 3.4 angstrom, 0.34 angstrom, 34 nanometer or 3.4 nanometer. It is 3.4 nanometer. Central dogma in molecular biology was proposed by Crick, Watson, Seck or Linnaeus. It is Crick. Circular DNA is present in prokaryotes, eukaryotes, viruses or mesokaryotes. Yes, it is prokaryotes. The number of BP base pair in nucleosome DNA 100 base pair, 200 base pair, 300 base pair or 500 base pair. Yes, it is 200 base pair. Transformation experiments were conducted by Korenberg, Hershey and Chase, Griffith, Liederberg. Griffith. Transformation was discovered in Streptococcus, Diplococcus, Tetracoccus or Staphylococcus. Yes, you are right, it is Streptococcus pneumoniae. DNA as a genetic material proved by Griffith, Hershey and Chase, Liederberg or Tatum. Yes, Hershey and Chase. Next, genetic material in TMV, tobacco mosaic virus and HIV viruses is RNA, DNA, proteins or amino acids. Yes, it is RNA. That's great. Isotopes used in semi-conservative method of replication is N14, P32, N15 or O18. Yes, it is N15. That's great. Semi-conservative method of replication was experimentally proved by Chargaff, Watson and Crick, Michelson and Stahl, Linnaeus. It is Michelson and Stahl. That's great. Semi-conservative method of DNA was proved by Taylor in E.coli, 5174, then Vicia faba or Streptococcus. Yes, it is Vicia faba. The main enzyme involved in DNA replication is DNA dependent DNA polymerase, RNA dependent RNA polymerase, DNA ligase or RNA polymerase. It is DNA dependent DNA polymerase. E. coli completes the process of replication within 40 minutes, 30 minutes, 28 minutes, 38 minutes. Yes, 38 minutes. That's great. In E. coli, the average rate of polymerization has to be approximately 2000 base pair per second, 1800 base pair per second, 2500 base pair per second or 2100 base pair per second. It is 2000 base pair per second. The DNA-dependent DNA polymerase catalyze polymerization in which direction? 3' to 5', 5' to 3', 3' to 5' or 3' to 3' or 5' to 5'. Yes, it is 5' to 3'. If replication is continuous, the strand is known as lagging strand, leading strand, Okazaki segments or discontinuous segments. It is leading strand because discontinuous strand पर Okazaki fragments का formation होता है. So Okazaki segments are joined by DNA polymerase, RNA polymerase, DNA ligase or cryptase. It is DNA ligase. Which of the following is used as an anchor for the addition of new DNA nucleotides? RNA polymerase, RNA primase, RNA primer, or DNA primer? It is RNA primer. That's good. Replication of DNA occurs in S phase, G1 phase, G0 phase, or anaphase. It is S phase. In eukaryotes, transcription occurs in nucleus, cytoplasm, endoplasmic reticulum, GC, Golgi complex. It is nucleus. The strand of DNA which does not code for anything is called coding strand, replicate strand, template strand, or antisense strand. It is coding strand. Transcription direction is 3 prime to 5 prime, 5 prime to 3 prime, 5 prime to 5 prime, or 3 prime to 3 prime. It is 5 prime to 3 prime. That's good. The promoter is located towards 5 prime end of the coding strand, 3 prime end of the coding strand, middle of DNA, or 5 prime end of the template strand. Yes, you are right. Very good. I'm proud of you. It is 5 prime end of the coding strand. The terminator is located towards 5 prime end of the coding strand, 3 prime end of the coding strand, 5 prime end of template strand, or no specific direction. Yeah, it is 3 prime end of the coding strand. Part of DNA coding for polypeptide is called recon, muton, cistron, or mutagen. Yes, it is cistron. That's great. Monocistronic RNA is present in prokaryotes, eukaryotes, mycoplasma, viruses. Yes, it is eukaryotes. Highly unstable RNA is tRNA, rRNA, mRNA, or hnRNA. Yes, it is mRNA. Then RNA polymerase 1 transcribes rRNA, tRNA, mRNA, or hnRNA. It is rRNA. RNA polymerase second transcribes rRNA, tRNA, mRNA, or hnRNA, yeah, DNA. Yes, it is mRNA. RNA polymerase third transcribes rRNA, mRNA, tRNA, or DNA. It is tRNA. Introns are removed and exons are joined in a correct order by splicing, capping, topping, or tailing. It is splicing or removal of introns. The genetic code is triplet. It was suggested by George Gamow, Morgan, Hershey and Chase, Hershey or Chase. So it is George Gamow. So one of the following is a homopolymer. UUC, CCA, CCG, or UUU. Yes, it is UUU. So the number of functional codons are 64, 61, 3, or 1. It is 61 because 3 codons are nonsense codons. Initiating codon is GUG, AUG, UAG, or UGA. It is AUG. That's good. tRNA is also called sRNA or aRNA, hnRNA, rRNA, or mRNA. Yes, it is sRNA soluble RNA. The secondary structure of tRNA shows clover leaf shape, S shape, L shape, or inverted omega shape. Yes, it is clover leaf shape. That's good. The amino acids in a protein is linked by hydrogen bonds, glycosidic bonds, peptide bonds, or covalent bonds. It is peptide bond. Translation is terminated by reading frame IF, initiation factor 3, CF1, or IF2. It is RF. It is repression, your repressor factor. So leg operon concept was proposed by Watson and Crick, Jacob and Monod, Liederberg Totem, या Beadle Totem. It is Jacob and Monod. The Z gene codes for, हम बात कर रहे हैं लैकोपेरॉन की. The Z genes codes for alpha galactosidase, beta galactosidase, permease, or transacetylase. Yes, it is beta galactosidase. Lactose is polysaccharide, disaccharide, monosaccharide, or trisaccharide. It is disaccharide. The Y gene codes for enzyme lactase, urease, permease, or cryptase. Yes, it is permease.